we're going to be dissecting an eyeball. And even though I'm generally an eye doctor for human eyeballs, there's a lot of similarities with cow eyeballs. So we've got our cow eyeball here. And one of the things to know about cow eyes is they're bigger than a human eye, which makes sense. But even though the cows are a lot bigger than humans, they're only a little bit bigger on the average cow eye. Average cow eye maybe be about 30 millimeters in diameter. Human eyes are generally average about an inch in diameter, which is 25.4 millimeters.
Sounds about right.
I think that's it. Anyway, so it's pretty close in terms of size. Believe it or not, human eye, despite you only see this much of it, is almost the same size as this. So there's going to be some similarities, some other differences. One thing that you can notice, we've already eliminated a bunch of the connective tissue that's all around the outside of the eye. And eyes have to last us our whole life. Essentially, we're not going to be able to survive, and a cow is going to have a much rougher time surviving if it loses an eye. The number one reason we have two eyes is so you have one spare. But at the same time, we don't want to make these things fragile. We don't want to make this something that's easy to lose or easy to damage in a permanent fashion. It's hard to go to the hospital when you are standing in the middle of a field eating grass. So one of the things you'll notice is that they're pretty tough. You can actually push on a cow eye pretty hard, even the center part. And don't try this at home, but human eyes are fairly similarly tough. And even though this is tough, the thing that it's stored within is your skull. And the skull has a nice bony socket all the way around it. That would allow the tough eye to potentially get squished up against the skull if it was to be struck. So what you have is a bunch of padding here, mostly fat-based, that allows a certain amount of give before, if you imagine like an egg, potentially being compressed. An analogy I like on eyeballs is that they're a little bit like a grape, right? It's got a pretty tough skin, and it's got spongy stuff on the inside, and it's kind of fluid. So it can be deformed by being pushed on, but it's not going to allow a whole lot of penetration. This area around the outside is more of a connective tissue. The whitish part is called the sclera, and it's actually kind of beneath a little bit of conjunctiva. But what you're seeing is mostly sclera, and that's the part that's not too painful on your own eye. This is fairly cloudy, and that's because of the preservatives used to help keep these eyes from going to mush. But you can see through it enough to see that there's a oval kind of a slit, which is different from the human eye. That's the iris. Think of that like a curtain that controls how much light comes in and out. Now, because of it's a slit, it allows more light in when it's in dim conditions. But under bright conditions, it doesn't get quite as small. So you're always going to have a little bit more glare. Cows, because there's something that other things can eat, want to be able to see well enough under low light conditions when things come out to hunt that they don't get surprised too easily. So that's part of the adaptation, the reason why they would have a vertical pupil, and cats will have even more of a vertical pupil, and other nocturnal animals similarly have eyes like this. Both cows and horses have similar kind of pupil sizes for you. One of the interesting things about why the cornea is usually clear, when I look up here at this model, you can see, of course, a clear cornea. Cornea is made up of a bunch of cells, and there's no other cells that are clear in our body. But because of the perfectly uniform orientation of them, as long as there's a certain amount of circulation and metabolism in this living structure, it does stay clear. And similarly, within the eye, which we'll get to shortly, the lens portion is clear, but it can become hazy as it sits. So very likely once we get in there, it's going to be fairly hazy, which we would call in humans a cataract, right? And that's the cataract, the lens and the cornea work together to help focus the light. Anyway, the one other thing we really want to point out here, can you roll this over for me, Bailey? Bailey's going to be assisting us by helping to manipulate this while I show you a couple different things. Right back here, you see kind of a post or a stalk coming out. That's going to be your optic nerve. And the optic nerve is the main pipeline between your eyes and your brain. The myelin is the part that you see, but the nerves are very fine conductors of electrical signals that go right down the middle of all of these myelin sheaths. And that same nerve cell is the same cell within your eye and back in your brain. So eyes can be considered the greatest extension of the brain, or the most exposed portion of your brain when you look at it. The other thing, if you turn it on its side, you should be able to locate about 4 different, here's one right there. And I don't need 2 of these. Hold on. There's one. If you keep rolling it over, You'll see a couple more. There's another one right here, and there's maybe one here. Those are the muscles. So those are the 4 eye muscles. And if you look over here on the human eye, we do have 4 that are prominent. 1, 2, 3, 4. And those are the same ones that the cow has. When it gets pulled, the eye looks up, down, left, and right. But the humans also have 2, one here, and one here. And those can cause twisting of the eye. Or for humans, if the eye is turned a great deal, now these become the up and downs. Because it changes the orientation of this globe. This is one of the subtle differences between human eyes, which should be able to scan and look more, versus a cow eye, which is fixed and on either side of the head, so they don't have to kind of work together as much. It's more, a lot of animals that are prey in this world do have to have a greater peripheral vision, and so they're able to avoid getting munched on, right? Okay, so what else we got going on here? Okay, the front portion that you see that's cloudy is the cornea. And then right beneath it is the iris. We'll be seeing those from the other angle shortly. And then we have 2 fluid-filled sacs inside of the eye. As Bailey gets going on this, go ahead and Bailey, I'm going to have you make an incision just behind the cornea, just posterior to the cornea. And then I want you to do it radially all the way around the outside. There's a specific technique for this, so I want you to watch how Bailey does this. It's a great way to keep you from severing your fingers. It'll get it started in there.
You see, this is what I should have done from the beginning, because we got right in.
Well, and feel free to change it up, just like folks should. It's harder to see what you're doing, but you're right. If the scalpel doesn't prove to be quite as sharp as we'd like, there's more than one way to open up a cow eyeball, apparently. Mm-hmm. So as Bailey's making that cut, realize that the cut on this model is slightly different, because it's lengthwise. That would be very difficult to do with an actual eyeball. What we're going to see is a lot of the fluid that's coming out you would think is from here, but it's actually more of the fluid is up in this area right in here, and it fills this front portion of your eye and the area around the lens. And that's all called aqueous humor. It's more water-like. Whereas the back is going to be more jelly-like, and that's going to be look a little bit like Jell-O that was left on a picnic bench for a little too long. So go ahead and take a look as she separates the 2 halves here. We got the anterior, or the front half, and you'll notice that the vitreous came out with it right there. So first, let's go ahead and look at, let's look at the front part of the eye. Go ahead and take that vitreous and so try to scoop it out gently, Miss Bailey. So in humans, it does become more watery-like as you become older and you go on in life. But this cow's eye is holding together pretty nicely the whole bit of vitreous there. Now the vitreous does adhere slightly to the lens on the back posterior pole. So she right now is doing a vitrectomy, which is something that happens for humans if they have really bad bleeding or leakage of stuff into the back of the eye. So I'm gonna set that down because that's now been removed. If you've ever heard of someone who has floaters, most floaters are bits of pigment that appear in the Jell-O of the back of the eye or the vitreous. So you can see that vitreous is, it's pretty, there's something to it right there.
Yeah, it's definitely
right?
sticking. And it's
yeah, it doesn't just scoop out like real
viscous.
easy. It's like the inside of a stress ball is kind of how I like to think of it. Now, we got a dome right in the middle, and of course that is the lens that we were talking about. That's going to be on this model, this right back here. Now you'll notice on the model, we've got all these little spiky guys. Do you see any little spiky guys associated with that lens there, Bailey?
Yes.
So let's go ahead and point at those. What we've got right along the edge here are all these zonules. And they're what hold, you can see when I do that, you see what's holding that lens into place? It's almost like a bunch of little cables. You guys can see those? And of course, since I'm pulling it pretty hard, you can see them start to separate a little bit. Now those cables, in a functioning eye, push and pull, or mostly pull or relax, in order to change the shape of that lens. So if they were to pull, and if the muscle that's right around here, called the ciliary body muscle, flexes, it pulls that lens and changes the shape. This right now, because there's very stiff curves, is going to be a very high-powered plus lens. But when you flex it, it becomes a little bit more of a minus lens or less overall power.
Right.
And therefore you can focus more at distance, or you can focus, for near. And that's how this lens shape changes. Now, as time goes on for humans, this lens becomes larger and larger. Every year, the lens has a primary function of helping us see, but has a secondary function of blocking high energy light, which is coming in the front of the eye. If you flip it over, it'll stay put. Although it might be slippery. So as sunlight comes through our eyes, or through the cow's eye throughout its life, it's going to be focused by the lens. But we don't want all of those light rays to get to the back of the eye. Because if it gets to the back of the eye, the problem we run into is UV damage, right? UV damage on brain tissue, like we talked about earlier, nothing that we want to deal with. So when we flip it back over and look here, the lens keeps adding layers, and eventually humans end up needing reading glasses. Cows don't have a lot of reading to do, so we probably don't have to worry too much about cow eye exams. So all of this you'll see is dark, and then if we kind of do a cataract procedure to remove the cataractus lens, you'll see something kind of interesting here. Right on top of the lens there, you see that pigment? That is where this lens, now probably not when the cow was alive, but this pigment that's right in there, right here and here, is where the iris touched the lens. And that usually doesn't happen until until you pass away, but it can happen to people, and that can cause problems like glaucoma, angle closure glaucoma. But I do see patients with this sort of pigmentation on their eyes as well. So let's go ahead and take this lens, and you can see that it's fairly easy to maneuver, although it's slippery. Not too tough, but certainly somewhat tough. We move that out of there. Sorry, I'm all shaky because I had too much caffeine at lunch. Okay, so now what we've got is a good view of the iris. This is the curtain that sits between the ciliary body and the cornea. And you can see it has that oval shape that we were talking about earlier. That's something that how cows are different from people. And with bright light, that curtain is going to draw closer, so you have less overall light get in the back. And in dim light, it's going to slowly dilate and become bigger. That's also controlled by the ciliary body muscle, by various parts of it. Okay, so now let's do something different. Flip that over and have it look back up at us. Now set her on the dissecting tray. One of the things I want you to try to do is try to see how tough your cornea is. How difficult is it to be able to penetrate this structure? Now, as you know, in humans, I said that this and this are responsible for helping you see. This is the structure that we operate on when we do laser surgery. And when they do laser surgery, they end up creating a flap here, but they don't cut through it. They cut into it and kind of peel it back, remold the shape of it to give you the prescription that you want with lasers that etch also microchips. Yeah, and then they lay it back down, and then it heals, and you're able to see pretty well. But we also talked about cataract surgery. Sometimes sun damage occurs and damages this lens over the course of your lifetime. And in order to help you see clearly again, they remove your lens by actually going through the cornea and making two little tiny cuts, like a little 2mm one and a 5mm one. And then they use a little mini vacuum with ultrasonic power to break up the inside of this and then scoop it all out, and then they put in a artificial one. And there's been a lot of technology updates on that. So after cataract surgery, would you guess that people see as well as they do after LASIK surgery? Well, the answer is pretty close. It's not, it takes a little bit more time, it's a little more invasive, there's a little bit more risks, but at a certain point in your life, remember, you're looking through a foggy windscreen. It's almost like an old car that's had a lot of rock chips or something on it, or even better, like plastic. You know, those headlights on cars that get a bunch of sun over time and then they look yellow?
Yeah.
That's what the lens looks like. And by removing that, you're seeing clearer already. And at the same time, they can put an appropriate power lens in there, and you can frequently get very good visual acuity or very good ability to see for humans. I'm not aware of them doing that for cows.
Not yet.
Okay, let's go ahead and head over to the back side of the cow eye. My guess is that this cow eye was hyperopic. I think he was a far-sighted cow. You know why I think so?
Why?
Because it looks like the distance to the back of the eye, to the front of the eye, it looks like it's not nearly as deep as we thought it would be, right? And if you're overly short with your eye, then you're farsighted. And if you're a little overly long with your eye, then you're going to be nearsighted. So farsighted cows, there you go. Once again, reading glasses for this cow would have been helpful. So we've got a couple different layers here. Now, after all this talk and all this studying of these eyes, you think about that brain tissue that we were talking about, you'd think it would be really substantial, like real easy to see. And look at it. It's just this little tiny tissue paper looking stuff. Now that's a retinal detachment right there. And you'll notice that nothing's holding the retina in place. It really is just pushed right up against the next layer down. And you can easily pull it apart. So why doesn't it just fall off in real life? Well, partially because of that jello sac that was back in there. So the whole thing is held together with this special sac that can push and hold everything in place. Now, go ahead and remove that the rest of the way, Bailey. You'll notice that as she peels it away, at a certain point it sticks. She can't peel it away. Where she's getting it stuck is right here. This pink layer is what she's removing, and it's getting stuck right here at the outlet port. Because like we said, those same cells that make up the retina are continuous into the brain. So you're seeing an area where we're going to have to cut them in order to fully separate the retina. So one of the things that's interesting is we were talking about how tough, and you guys have been cutting into these eyeballs, you know how tough these eyes are, but the part that so many people think is the most important part, or the most magic part, or the most special part, and they got 10 layers and all these specialized cells, we do these electron microscopes and stuff on them, they really are so very, very fragile. It's just held in place. Now, I think it's funny, any structure in the eye is no more special than the other one. They all need to function together in order for things to work. But it's interesting that we spend a lot of time talking about the retina because it's a pretty specialized structure. And now you can see, like, if there's problems for a human retina, how easily they can become a bigger problem. One little hole allows things to peel all the way off. These little retinal tears is what we worry about for humans. Now, when we're looking behind the retinal tissue, we now have this little shiny area right back here. It almost looks like the inside of an oyster. That's called the tapetum lucidum. That's not a zone that we as humans have. This is another one of those subtle differences between humans and a good number of animals. You'll notice if you shine a bright flashlight at a cow's eye at night, you're going to get a greenish kind of a view, and that's this. That's the light bouncing into the eye and then bouncing out, and that's what you're seeing. And various animals have slightly different colored eyes. But you'll notice that humans, what color do they have?
Red.
Yeah. So when you take photographs, you'll often see red eye, right? And that's because you'll notice it more in dim light because your pupils get larger and then more light bounces into the back of your eye and then it's reflected back as red because the back of the human eye is pinkish, orangey red. So that's kind of what you're looking at when you see that. They have this layer so that they can collect, almost like a radar dish, more light and more effectively work in low light conditions. So even if there's just a light starlight, they can gather that up a little bit more effectively because it's a little bit like a mirrored surface and it helps collect more of that light like a radar dish. Now, humans don't have that because under bright light conditions, where humans are generally superior in their vision, this causes more glare. It makes it harder to see clearly. They wouldn't be able to see 20/20. So cows never see quite as clearly as we do, but they're a little bit more sensitive under low light conditions. So go ahead and as we try to kind of remove that, you can try doing that. There you go. That layer is then right on top of a darker layer, or continuous with a darker layer. And that whole layer right there is the next layer down. It's a very vascular region in the eye. That's the kind of the blood supply that allows all of the retina tissue to soak in the nutrition from your bloodstream. So there's a lot of blood vessels and stuff in there, small ones, tiny ones. And you could think of it almost like a capillary bed, almost like a nice thickened region of vascular tissue, I suppose. So its main function is to supply nutrition to the entire retinal tissue in the back of the eye. Because you'll notice that if there's all this clear stuff, you can't have veins and arteries flowing around in there. Now, there are a couple veins in the that you may be able to see when you, as we did the retina part of it. There's some areas that you can see, like right in there. You see that? That might be part of the vasculature in the back. This model is not very good because it shows just tons and tons and tons and tons of blood vessels. That's not That's not typically what we see. Usually it's a couple large branches, and then it goes into a bunch of smaller branches. Okay, so we've got the zonules and all the lens stuff. We've got the vitreous, we've got the muscles, we've got the iris up front, and then we've got the cornea. Last things to know. Remember when we looked at the outlet on the back and we saw the optic nerve? That optic nerve travels straight through to right where all the retina was attached, right here, where we still have a little bit of a nub here. So that's your main pass-through. One of the concerns for human beings is if the pressure in the eye builds up. The pressure gets high because too much of that aqueous, that watery stuff is made, and it pushes against, and it doesn't drain quite as, usually it drains right inside here. So if we can do this, this would be real tricky. See if you can make an incision right on the edge of the cornea in order to see if we can separate the cornea from the front, and we can look in there. I'll talk more about glaucoma as we go. Glaucoma is going to be generally an outflow problem, and the outflow is what she's going to be able to show you once we get into this front anterior chamber of the eye. And that's truly the name that we use when we talk about ocular structures. You don't have to go quite that, yeah, you can go up into the cornea about like 2 millimeters, like up this way. Right around there. Yeah, because if you're too close to the root, it ends up being really, really tough to get in.
Right.
Now go ahead and try to see if you can, oh, this is gonna be great. Now that is about as large as the cut that they make to go in for cataract surgery. And let me show you something else that's kind of neat about that. When they make that cut, they actually do it with their knife, and they go in like this, down, over, down again, which is kind of a funny cut. But that's because they found that the internal pressure of the eye, when you pull your knife out after that kind of a cut, seals itself off, and you don't need any stitches. You could push and have a little bit of fluid leak out, but if you cut it just right, that's how they do these surgeries without stitches nowadays. And they make a larger cut like this, and they make a really small one up here. We're not going to make you do that. And then they use those two. They have a pokey that goes in one side to help manipulate, and another that goes in, and that's how they do cataract surgery. Go ahead and keep working on that cornea. Jumping around to glaucoma again, which was that pressure disorder. The reason that you're worried about too much pressure in the eye is the only real outlet is, like we said, back in here. It's that optic nerve. And if the internal pressure is pushing down too hard on that optic nerve, well, imagine if the pressure is pushing down on the optic nerve, you can fray these delicate wires that are going over this firm structure. This outer bit, this sclera, this white part, is very, very strong and protective, but if you're pushing too hard, you could pinch those nerves. Anyone who's ever hit their funny bone will know how much nerves don't like pressure. Right? So let's take a look at that corneal tissue. Believe it or not, as much as everyone loves to talk about the lens of the eye, the lens only does actually about 40% of the focusing of light. The cornea does more. And that's not because it doesn't change shape very often. What it does do is it light is traveling through air and then it goes into fluid, and because of that transition, this ends up being a more potent lens, and that's why LASIK is so very powerful. Now you can see that's pretty tough. Like you could try tearing that with your fingers.
Really thick.
And it is strong stuff. Even though it's clear and you would think it's super fragile, and let's be fair, none of us want to have a damage to our cornea, it is really tough.
Yeah.
So tough, in fact, that the teachers at the optometry school want to show us how to remove stuff from eyes, but they want to show us how to identify like scratches and damage to corneas, and they used to try to do it with cow eyes, but they had such a hard time trying to make something that you'd see. They were like throwing them and skipping them off of like sidewalks and stuff to try to get them scuffed up, and it doesn't, it's like, you want to take a sander and like, eyes are tough. And what's interesting is eyes can replace their tissues. So as long as you don't get down too deep in the cornea, it can repair itself, and that's also how LASIK works. Now what we're looking at here, this is awesome. Man, this is great. I'm so psyched. Check this out. This is the area right in here we call the angle. Okay, I'll show you from this side. Right underneath here, back in there, is where the fluid drains for the eye. And when we talk about glaucoma, usually that's getting plugged up somehow. They've got a variety of very fancy treatments for this. Mostly it ends up being a drop to help open those vessels up and drain more of the fluid from your eye so that pressure doesn't build up. And as we were talking about earlier, remember when we looked at this lens here with the stuff that was stuck to it? You can look here. Imagine if this, which is the area that all the fluid comes up and out of, if that was stuck to the eye and doesn't allow any fluid to come out, wow, that pressure can go up really quick. That's called angle closure glaucoma. And you know how we get worried about pressures like 25? You're talking 60, 70, like, oh my gosh, your vision starts to black out. At a certain point, the pressure is so high that blood flow can't go into the eye, and it's like you got 30 seconds, you know, to a couple minutes before you get permanent damage to your eyes.
Yeah.
So that's partially why we always want to check on these things as human eyeball doctors. Anyway, looking at here, we've got, that would be one of the regions that we like to examine. Because of optics, it's really hard to actually see directly into there without lenses that rest directly on your eye. That's kind of wacky. Anything else I could think of? Okay, so we talked a little bit about glaucoma. We talked a little bit about cataracts, right, which would be this guy here. We talked about laser surgery, right? That's some other things that people, you know, it's helpful to know about. And on the back of the eye, there's all sorts of retinal detachments that we talked about, or full retinal loss and full vitrectomy. I don't think that this patient's going to be recovering, but then again, we all are smarter for the sacrifice of this creature. The last thing to know is the retina in humans, as time goes on, can actually have this darkened area leak fluids that slowly seep up through the retina. Not like the nutritious fluids, just fluids that essentially cause deposits to form. And that's what we call macular degeneration. Especially if the fluids are coming out pretty quickly, we can cause problems there. Last thing to know, with all the things going on in this eyeball, it is the one place that we can see living brain tissue in a living human being. It's also the only place you can see living blood vessels, because there are those branched blood vessels that we're going to be able to look at. So we look for things like, you know, systemic problems that would be occurring elsewhere in your body, but you wouldn't be able to see, right, if there wasn't, you know, if you can't see it live. And so if you see damage in the back of the eye from things like diabetes or cholesterol, well, you can tell that that's also happening for the rest of the body. I didn't get too much of a look at our cow, so I'm guessing he didn't go down from cholesterol, but
Maybe.
yeah, actually, I don't think that's possible for a cat.
Well.
We'd have to talk to him a bit.
We'll see.
Any other thoughts or questions, gals? What do you figure? Does it seem like a...
Did you go over the choroid?
Yeah, choroid is that vascular. It's that darkened vascular structure right there. Sorry, that's the name of it. It's the choroid. I think we're all set there.
Good.
If you have any questions or comments, feel free to direct them to Bailey, who you can reach through our website.
I am an expert.
Awesome. All right, guys. Have fun with your dissections.